go ahead and get started. So I'm going to introduce Joe Smith from University of Washington. You know, we often do these little introductions. You start by Googling someone, and when your name is Joe Smith that you're Googling, it's a little imprecise as to what you will get. So the bulk of the responses from a Google search are Joseph Smith, the founder of the Mormon Church. Many, many pages of that. And then you get this University of Washington affiliate associate professor in global health, University of Alabama department chair in political science, the vice president of a finance company. I suppose I could actually make this a presentation, couldn't I? Baseball pitcher, a current one. Former pro basketball player with a long career. The Oregon district attorney for Umatilla County, who is also the chair of the Oregon Democratic Party and gave a TED Talk on paper towel use. There's an actor with rights to the joesmith.com URL. And if you add fish to the query to make it a little more precise, there was a Joseph Smith Fish, who was also Mormon and settled early in Utah. A graduate student at the University of Montana who used to work for a Fish and Wildlife department. Another NOAA scientist at the NIMS lab in Beaufort, North Carolina, studying menhaden. And then we get this Joe on page 2 or 3, depending on how you do the search, with fish in the title. So Joe got his master's at Eastern Washington University, completed a PhD at the University of Massachusetts Amherst in 2012, and he's going to talk about his current research at University of Washington as a postdoc with Tom Quinn. Research interests include causes and consequences of fish movement within ecosystems and sort of the stuff around that. So I'll let you talk.
Great, thank you. Thanks, Kinsey. And thank you all for coming today. And I'd like to thank David Huff for inviting me to come give this talk today. So the title of my talk is Examining the Ecology and Predation Impacts of Non-Native Fishes in the San Joaquin River, California. So this is a large project that's a collaboration between the Southwest Fisheries Science Center and University of Washington. And I want to start off by just introducing and thanking the large group of people that are a part of this project. So up in the top left, we have Steve Lindley, who's the director of the Fisheries Ecology Division at the Santa Cruz Lab. Sean Hayes, who is now at Woods Hole, Massachusetts, in the Northeast Division. David Huff, who is now the chief scientist at the Hammond Station in Oregon. Cyril Michel, who really leads our team down in Santa Cruz Lab for field research, and then Tom Quinn, who is my postdoc research advisor. The second team that I'd like to introduce is our hydroacoustics team. So these are all from the Southwest Fisheries Science Center down in La Jolla, California, and includes Dave Deemer, Randy Cutter, and Suzanne Manoujian. And so they really led up the hydroacoustics effort, which I'll just give a brief overview about in a little bit. And then our field research team. So we had a large group of people that were able to come out and help over the 2-year period that we did this study, and without them, we couldn't have done this. So a quick outline of what I'd like to talk about today. First, I'll give you some background on the study area and our study questions. Then I'll get into the specific study objectives and then work through what we did. So that includes looking at the distribution of predators with electrofishing, hydroacoustics, and telemetry. We looked at predator diets using genetic barcodes and then used those to look at some bioenergetics. And then last, I'll talk about our predation event recorders and our use of a before-after control impact design for predator removals, and then how these predator events were related to environmental covariates, and then follow up with some conclusions. So a little bit of background on the system. We did our study in the San Joaquin River, which flows from south to north, and that meets up with the Sacramento River, which flows from north to south. And they flow into the San Francisco Bay and out of the under the Golden Gate Bridge. So this system used to be heavily dominated by wetland habitat. It was very highly connected dendritic system, and now it's very heavily modified and very dominated by agriculture. In 2014, an NGO, the American Rivers, designated the San Joaquin River the most endangered river in the United States. And because of this, it got a little bit of press from CNN. One of the reporters decided he wanted to go on a trek from the headwaters in the Sierra Nevadas to the Golden Gate Bridge. And so he actually has a blog that where he documented this adventure trying to get to the Golden Gate Bridge. And as you notice, there's a big chunk of the river that dries up basically every year. So a lot of the water from the Sierra Nevadas never even makes it to the Golden Gate. On top of other issues, California has been in a very large drought. So this is a plot of drought through time with the percent land in drought. And so we can see a few droughts since the year 2000, and we're kind of at the tail end of a pretty significant drought here recently. So I put a box around when our study period was in 2014 and 2015. So we were right in the middle, basically, of a pretty significant drought. So in this system, there are endangered and listed species, including Delta smelt, Chinook salmon, and steelhead salmon. And over the last decade or so, there's been a very low survival of salmon smolts emigrating out of the system. So we really want to understand what is causing that low survival. So there are many reasons for low survival, but one thing that people really point to is the effect of alien, non-native fish predators. So because people are interested in that, a report was commissioned in 2013 to specifically look at predation effects on salmon. And here's just a quick quote from that report. It said, given extensive flow modification, altered habitat conditions, native and non-native fish and avian predators, temperature and dissolved oxygen limitations, and overall reduction in historical salmon population size, it is not clear what proportion of juvenile mortality can be directly attributed to fish predation. So this kind of gap in knowledge led to the study that we conducted. We're really trying to look to see if we can quantify fish predation in the system. So who are the fish predators we're talking about? We focused on the 4 most abundant fish predators in our system, which included striped bass, largemouth bass, white catfish, and channel catfish. And so these maps in brown show the native distribution of these fish, and all the blue points are places outside of their native range where they're found. All of these fish were introduced in the late 1800s to the system, so they've all been in the system for quite some time. So now I'll go over our specific study objectives. Our first objective was to determine the abundance and local spatial distributions of fish predators. 2, we wanted to quantify the magnitude of predation on salmon smolts with genetic analysis of predator stomach contents. 3, manipulate the density of predators to assess the influence of predator density on the predation rates of salmon smolts. And then 4, determine how predation on salmon smolts may be influenced by physical habitat, water chemistry, and other environmental features. So here is an aerial photo of the region, and you can see San Francisco Bay on the left and Stockton, California kind of highlighted to the right, and the red box outlines sort of the southern delta area. So now I'll zoom in on the red box, and here is the delta, and if you look up in the left corner, you can see Sherman Island. That's near where the Sacramento River is coming into the system. The bottom left shows Clifton Court. This is the area that they collect water to send to Southern California. So this is a very important area for water. And Then on the right, we see the red box with the San Joaquin River, and that is our study area. So I'll zoom in to that now. So here's our study region. It's about 25 kilometers long, and the river is flowing from south to north. We broke the region into 3 blocks, and within each block, we have 3 1-kilometer study reaches. And within each block, we have a control reach, a removal reach, where we would go in with electrofishing and remove as many predators as possible with multiple passes. And then we would move those predators from the removal reach to the associated addition reach within that same block. So we have 3 replicates of 3 different treatments. So our first objective was to look at the distribution, abundance, and local spatial distributions of predator fish. So for this objective, we used 3 different methods. The first method was multi-pass electrofishing surveys. So here is a combination of all fish combined, caught, and here are the relative abundances of those predators. So we see it's dominated by striped bass, followed by largemouth bass, then white catfish, and channel catfish. And then the other species make up a small fraction. This table shows the number of predators that were removed in each of our removal reaches for each year. So 2014 and 2015, and then combined. For a grand total of about 2,872 predators removed over the 2 years. So at each of our removal reaches, because we did depletion estimates with electrofishing, we were able to get population estimates for that reach, for the abundance of fish. So here are our estimated abundances of striped bass in each of our removal reaches in 2015. So we see at removal reach 1, which is down here, we have an estimated number of about over 1,000 striped bass within this 1-kilometer reach. If we look at the other reaches, we see a much smaller abundance estimate, and I'm going to show you some telemetry data a little bit later on striped bass, and we see that they are very highly mobile and very patchy in their distribution. So we see a patchy distribution and big differences among reaches for striped bass. Now, if we compare that to largemouth bass, with largemouth bass, we were actually able to look at abundance estimates using 2 different methods. One was with e-fishing as before, but also with mark recapture using pit tags, which are the blue bars. And surprisingly, with 2 different methods, we came up with pretty similar estimates of numbers of fish in each of the reaches. And as you can see, there's a pretty even distribution of numbers of fish at different reaches, around 300 fish per 100 kilometers in each of the reaches. So pretty even and consistent distribution of largemouth bass. Due to electrofishing techniques and collection efficiency, we weren't able to get estimates of catfish in the system because of sort of the bias in collecting fish with electrofishing methods. So the second way we looked at the distribution of fish with hydroacoustic surveys. This was our La Jolla team that did this work. And in the upper left, you can see the boat with all the high technology that we used for these surveys. On the right, you can see some cartoons of the boat, and looking towards the bow, you can see how the different sonars, how they come off the boat, and then a bird's eye view in the bottom right corner. So with the hydroacoustic surveys, we actually get 2 really cool pieces of information. The first thing we get is really highly detailed bathymetry of the system. So we can look at the depths and differences in depth throughout the river. The other thing we get is fish targets. So the fish targets are the little dots that you see throughout the river, and the target strength of those dots can be used as an indicator of the fish size in the system. So we can look at fish through time. So here on the left, we can see all of these little tiny dots are actually fish targets in the system. So this is removal to the middle reach. and this is the detailed bathymetry that we get from the hydroacoustics. So there's some really interesting patterns where we see these scour holes, and the numbers of fish overlaid on top of those scour holes really kind of overlap. So some pretty interesting hot spots of fish in the system. Our third method of looking at the distribution of predators with acoustic tagging. So we had kind of two main objectives with this. We wanted to examine the differences in movement and residence among our 4 different predator species, and we also wanted to look at the effectiveness of relocation. So we removed predators and we moved them to the addition locations, and so we wanted to see how quickly would those fish return to their capture location. So we were able to do that with a subset of tagged fish. So the table shows the number of predators of each species that we collected in each year and the number that were transported, and so we had a total of 300 fish tagged over 2014 and 2015. So again, here are our study reaches, but on this plot you see these little yellow gates. So each of those little yellow lines indicates 2 acoustic receivers, and so they basically work as gates in the system. So every time a tagged fish swims past that location, the unique ID of that fish is identified and the time at which it passed. And so we can look at movement between the areas and the amount of time spent between each of those gates. So for this talk, I'm going to focus on just the fish that were captured at removal 2 and then moved to addition 2 here. And so also I want to point out that river kilometer 0 is what we're calling up here, and river kilometer 25 is down here. I'm going to show you a series of plots where river kilometer 25 is going to be at the top and river kilometer 0 will be at the bottom. So it's going to flip what you're used to seeing here in just a second. So again, fish captured at removal 2 and moved to addition 2. So here are the plots that I'm going to show you. So again, we have river kilometer 0 and 25 here and time on the x-axis. And all of these fish were caught at removal 2 with this red line and then moved to the addition 2 here. So first I'm just going to show you one straight bass. So this bass was moved here. It headed downstream and spent quite a bit of time out near the bay and delta. It came back into the system late June or late May, spent some time in the system, headed back out to the bay, and had a series of back and forth movements kind of throughout the summer, spent some time in the system in late August, around August, and kind of headed back out of the system. So that's just one fish, and now I'm going to overlay 9 more fish on top of that. So here we have a total of 10 fish, and you see a lot of back and forth movement throughout our entire 25-kilometer study reach, and a lot of time spent out towards the bay. Now I'm going to contrast that with largemouth bass in the system. So here are 10 tagged largemouth bass that were also caught and tagged and released at the same location. So you see a lot less movement throughout the system and a lot of site fidelity for their capture location, a lot of them spending a lot of time near that location. Now, looking at channel catfish, we see not as much movement as straight bass, but maybe a little bit more than largemouth bass. And then finally, white catfish. We see, again, some movement, but probably not to the extreme of straight bass and not as little as largemouth bass. So we just recently got in, this is data from 2014, and this is just the first part of 2014. We just recently got all of our data downloads a few weeks ago, and so hot off the press, we can see how these fish changed throughout a 2-year period. So now I'm showing you straight bass at the top and largemouth bass at the bottom, and now we have 2 years of data. And the black lines are fish that were tagged in 2014, and the purple lines are fish that were tagged in 2015. So again, at the top for straight bass, we see really high movement throughout the whole system, Again, we see that in 2015, but interestingly, the fish seem to disappear throughout, not when they're not during the spring. So if they're there in the spring, but they don't seem to be there the rest of the year, especially in 2015. Now, if we compare that to largemouth bass, you see that they have, again, this really high site fidelity to their capture location. We see a little bit more movement in 2015 and 16, but again, you see this really high fidelity to their capture location. So the other way we can look at this is the proportion of time that they spent within each of the gates in each of these regions. So these, again, here are striped bass. Here's their capture location and transport location, and the percentage of time on the x-axis, and each of the gates here on the y. So here we have in red the transported fish, and in black are the non-transported fish. So those are fish that were captured and released at their capture location. So what we see here is we see that fish kind of spent a lot of time in a lot of different regions throughout the area, and we do see a little bit of difference between transported and untransported fish. So little region fidelity for striped bass. Compare that to largemouth bass, and we see a high region fidelity for their capture location. So they spent a lot of time at their capture location. So pretty stark difference between the two species. Channel catfish, we see that they did not have necessarily fidelity to their capture location, but they did find a region and spend a lot of time there. So some region fidelity, but not necessarily the capture location. For white catfish, we see something pretty similar to that, where they might have region fidelity, but not necessarily to their capture location. All right, so the last question we wanted to ask with the telemetry data is, how fast do these fish return to their capture location after being moved? So we have return time in days on the y-axis in our different species here. So first of all, just how many of the fish that were tagged actually returned to their capture location? So we had 63% of channel catfish, 69% of largemouth bass, 44% of striped bass, and 74% of white catfish ended up returning to their capture location. And if we look at the overall mean among all the different species, it's about 8 days. And this is actually pretty surprising, since the addition location was only about 2 kilometers away from the removal location. So if these fish were highly motivated, they could probably be back to their capture location within a matter of hours. So here are box plots showing the distribution of return times. We had fish returning as quickly as within the day, but we have these really high times of up to 20, 30, or 160 days after they were moved, where they finally make it back to their capture location. All right, so now I'm going to switch gears and talk about quantifying the magnitude of predation of salmon smolts with genetic analysis of predator stomach content. So we collected predator stomachs in both years, and when we collected stomachs, it is kind of important that we collect fish during the time when salmon are actually in the system. So every year, the California Department of Fish and Wildlife and the IEP go out 5 days a week throughout the smolt emigration season and tow a trawl. And from that, they get estimates of salmon smolts in the system throughout the emigration season. And so these are plots, so through time of the emigration for each year, and the red line indicates the times in which we collected diet. So we did pretty good at collecting fish, at least when there were salmon in the system. So we collected about 400 diets per year with a total of 837 diets. This lab work was done by Kramer Fish Sciences, and it tests for the presence or absences of species of up to 12 prey species in the diet. And the amount of time that the DNA stays in the diet is around 2 to 3 days. So that gives us about a 2 to 3 day window of detection for the DNA. So these are the 12 species of fish that we looked for in the diets of our predators. But to today I am just going to focus on the Chinook salmon in the diets of the predators, but we have the data for the other species as well. So we have 2014, 2015, the frequency or the number of diets, the proportion of diets in the stomach of each of the predator species. So first, 2014, surprisingly, we found the highest proportion of diets were from channel catfish, with 26% of the diet having the presence of Chinook in their diet. Largemouth bass had 3%, striped bass 6%, and white catfish 3%. So all of our predator species detected Chinook in their diet, and channel catfish had the highest proportion. In 2015, we had similar results, again with the highest number for channel catfish, and the second highest was white catfish, and 1% for largemouth bass and striped bass. So you saw this before. This was the population estimate of striped bass, and I'm just reminding you that it's kind of patchy distribution. And then you saw this before with largemouth bass, and we see a pretty even distribution. So because we have estimates of the number of fish, and we have estimates of the frequency of occurrence in the diets, we can use bioenergetics to estimate the number of salmon smolts eaten within each of the reaches. So here's an example of that. For removal 3, on the x-axis, we have the estimated number of predators in that reach for 2 years, for 2014 and 2015. And then on the y-axis, we have the estimated number of smolts eaten over about a 2 to 3 day window, because that's what our genetics can detect. So with largemouth bass, the lines indicate the estimate, and the shaded area are our confidence intervals. So in this region, we see we estimate about 5 to 10 salmon smolts per kilometer per two days for largemouth bass. And because largemouth bass were pretty evenly distributed throughout the season, throughout the study area, we could extrapolate this out to most of the study reach of, we would estimate about 5 to 10 smolts eaten per kilometer of the reach. Now, if we do this for striped bass down at removal 1, and remember that's where we saw the high abundance of striped bass, we get these estimates. And so here we see estimates of around 25 to 100 smolts per kilometer per 2 days. But I want to point out that the distribution of striped bass is very patchy, and with the movement data, we saw that they move a lot through time. So this estimate probably wouldn't be able to extrapolate out very far compared to other reaches in the system. All right, so now moving on to actually manipulating the density of predators to assess the influence of predator density on predation rates of salmon smolts. So I mentioned to you that we removed as many predators as we could using multi-pass electrofishing. We used 3 electrofishing boats that simultaneously shocked, and we did anywhere from 5 to 7 passes throughout our study reaches. So here are the proportions, the relative proportions of predators that were caught in each of the reaches. Here again, we see that it was dominated by striped bass down here at removal 1, and dominated by largemouth bass at these other 2 regions. And just a quick reminder of the number of fish that were removed in each location in each year. So in order to evaluate the effectiveness of removals, we needed a way to evaluate predation. So the folks down at the Southwest Fisheries Science Center developed what we're calling the predation event recorders. What these are floating pieces of PVC, and at the top there's a GPS collar, and on that collar, so with that collar we can track them as they float down the river. At the bottom we have a live tethered salmon, Chinook hatchery salmon, attached to a fishing line which is attached to a magnet. That magnet is attached to a timer. If the fish gets eaten, it pulls the magnet and starts the timer. So then when we retrieve it, we can match the timer time with the GPS time and get exact locations of predation events. We also have a GoPro camera, which is watching all the action, and we can identify which predator fish actually ate the salmon. And on top of that, we have a temperature and a light intensity logger as well. So here is what those look like. Here's one floating in the water, and here's one out of the water, and a cooler full of salmon smolts. So how we sampled is we would take a boat to each of our locations within a block within a day. So one boat would go to each reach, and we would simultaneously deploy 10 tethers, and then we would let them float for about an hour. And then each of our study sites were sampled at least 3 times before removal and addition efforts, and then at least 3 times after removal and addition efforts. So here is what the data can look like. So each colored line is a track as it floats down the river, and every red dot indicates a predation event. So you can see just by looking at this map that we actually have hot spots of predation occurring up here near the scour hole. So now the fun part. Here's some videos. So this first video, I'm going to show you actually 4 different videos. This one is a montage of predation events, and it shows it being deployed and retrieved. So it's going to, oops. All right. So here's predation event recorder being deployed. And the first thing you'll see is the smolt swimming around. So you can see it swimming around up here. Again, here's swimming. And here's some large carp swimming by. And now here's going to be a montage of predation events. So now I'm going to, because those were pretty quick, I'm going to show you a few more. So here's going to be a few striped bass predation events, and it's going to show you in real time, and then it's going to show you in slow motion. That was real time, and that'll be slow motion. So you can see we can actually do a pretty good job at identifying the fish. Here are some largemouth bass predations. Initially, we didn't have high-resolution video, and it was very difficult to actually slow it down and see well enough. But with these high-definition videos, we can actually get a pretty good idea of the predators. And then finally, some catfish, just in case you don't believe that they'll eat live fish. So some pretty cool footage there. So one thing we wanted to think about was the time of day at which we deployed the predation event recorders. So in 2014, we sampled through time, and you see hour of the day here on the X and the percent predation on the Y. And you can see really a tight link between the percent predation and sunrise and sunset. So in 2015, we standardized all sampling so that it happened at around sunset every night. All right, so to the big question of do removals or additions impact predation event recorder predation? So here we evaluated this with a Cox proportional hazards model, and these plots have time on the x-axis in seconds, and then the proportion left alive on the y-axis. And so we have our before and after, and then our control, removal, and addition reaches. So first of all, for our controls, we have our before in black and our after in red, and we see not much difference between the two, and these are 95% confidence intervals, and they're widely overlapping. So no difference at the controls, which is what we might expect. At the removals, we see lower survival before and a little higher after removals. But you can see that these confidence bars are widely overlapping, so no significant difference. And then with additions, we have, again, not much difference afterwards. So we see no real clear effect of removals or additions in the system. And this is kind of what someone might expect, because it's an open system, and these are really big reaches. And there's plenty of predators just outside our removals that can move right back in after we remove them. All right. So now, lastly, I'll talk about determining how predation on salmon smolts may be influenced by physical habitat, water chemistry, and other environmental features. So to do that, we deployed these, and we measured environmental covariates at the same time. So we measured temperature, light intensity, water velocity, conductivity, turbidity, tide level, bottom slope, the density of fish targets from our hydroacoustics, depth variability, distance to shore, depth, time to sunset, predation event recorder speed in the system, the distance to pools, and the site ID. So in 2015, we had over 1,000 deployments and over 360,000 observations from those deployments. And so we used, again, a Cox proportional hazards model and model selection with AIC to see which covariates influence predation. So one of the big differences that we saw was differences in survival among our study reaches. So again, these are the plots that you've seen before with time on the X and proportion alive on the Y. And what we see here is these three dotted lines correspond to the reaches in block 2. And we see that the highest predation occurred at control 2 compared to all other reaches. These three dotted lines at the top correspond to those in block 3, and A3 is the reach that had the lowest, or the highest survival and the lowest predation. And then we have 3 lines here that indicate block 1, which had sort of intermediate survival. So big differences between study reaches. From our model selection, we ended up with these variables, which were significant in the analysis. So each, the variables in green had a positive relationship, so 1 unit increase in the covariate was increased survival, and those in red mean that survival decreased with a unit increase in the covariate. So now I'm going to quickly work through each of these different covariates. So again, these are the similar plots that you've seen before, and I'm going to show 6 lines on each plot. The skinniest line is the minimum value of depth. The thickest line is the maximum value of depth, with the first quartile, a median, a mean, and a third quartile in between. And the median and mean are in bluish color, if you can see it. So here's what we have for depth. So we see that the highest survival was at the lowest depth, and the lowest survival was at the lowest depth. So depth influenced predation. Now switching over to water velocity, again here negative values indicate a flooding tide water velocity, and positive values are an ebbing tide velocity. And we see that predation was highest at ebbing, fast-moving ebbing tides. Time to sunset, which I talked about a little bit before, was also very important. So if we look at exactly sunset, 0 minutes to sunset, this red line, that showed the highest predation. The negative minutes indicate time before sunset, so 191 minutes before sunset had the highest survival, and 80 minutes after sunset had kind of intermediate survival. So we kind of see this U-shaped pattern. Light intensity also had impacts on survival, so low light conditions had the highest amounts of predation, and really high levels of light, basically daylight conditions, had almost no predation. Tide level was also important, so negative values are low tide, positive values are high tide, so at the highest tides we had the highest amounts of predation. Turbidity was also important, so higher turbidity had higher predation, although I do want to point out that 18 NTUs is not really turbid. Here's 25 NTUs, so relative increase in turbidity, increased survival. Then temperature, we have the lowest temperatures had the highest amounts of predation. And then the last variable was distance to shore. So the closer they floated to shore, the more predation, and in really wide areas when they were really far from shore, we had the lowest amounts of predation. So with that, I want to give you a quick summary of everything I've talked about today. First, I showed you that largemouth bass were uniformly distributed among the study reaches. Straight bass were really patchy in their distribution. Hydroacoustics really helped provide us with high-resolution bathymetry and gave us fish density estimates through time. I showed you that striped bass are highly mobile and can move a lot throughout the system, and largemouth bass move much less and have high sight fidelity. Catfish had intermediate movement between largemouth and straight bass. Channel catfish had the highest frequency of Chinook in their diet. And with the combination of the diets and the abundances, the population size, diet, frequency, and movement patterns of predators can interact to result in varying impacts on salmon. So I showed you the differences between largemouth bass and striped bass and how their potential impacts on salmon smolts, and how it really can vary depending on the population size, their movement patterns, and their diets. We found no clear effect of removals or additions in the system. And finally, the per predation varied among sites and was highest at ebbing high tides near sunset in deeper, faster water with lower temperatures, higher turbidity, and floating closer to shore. So with that mouthful, I'd like to thank everyone that's helped out on this project, a huge group of people, and I'd like to thank California Department of Fish and Wildlife, who did the electrofishing, and the California Department of Water Resources, who funded this research. And with that, I'd be happy to take any questions. Thanks.
It's a microphone for questions so that they can hear on WebEx.
Hey, Joe. Nice to talk.
Thank you.
Given all those correlations you showed with the PER data or PER information, what do we know about the natural migratory behavior of salmon going out?
Right. So there is some evidence that they move more at night than the day, and they do tend to, I think, be closer to the surface as well. So there obviously is a sample bias with the predation event recorders, but there's a sampling bias with any sampling gear. So electrofishing samples fish differently. But if we can do basically the same thing in different areas, then we can kind of compare the relative predation rates between each of the systems. So yeah, there is some bias there in compared to what would naturally be happening in the system.
Hey. I had a question about the 2 years in which you sampled diets, because it looked like the second year had many fewer diets that contained Chinook salmon than the first year, and maybe that's an artifact of sampling. But if it isn't, then is that attributable to any other broader factor that you've thought about?
Yeah, so the diets actually were not too different between the 2 years. They were pretty, so the big difference is in the first year we saw about 26% of catfish diets that had Chinook in the diet, and then in year 2 we had about 8% of diets. But because actually channel catfish are one of the lesser abundant species, and so we couldn't capture as many of those. So that's why we also had really high standard error bars for those estimates. So actually, if we look at the standard error bars between years, they actually are pretty comparable within the range. The other species of fish were actually fairly similar, also within kind of the standard error bars between the years. So it's interesting. So we saw this really potential high level of catfish in the diet, Chinook in the catfish diet. But when you look at the overall population size, at least from our electrofishing, catfish were much less abundant in the system. I did mention that our capture technique was potentially biased for catfish, so maybe there are a lot more catfish in the system than we really know about. But if in fact striped bass and largemouth bass are the dominant predators in the system, their population sizes are much, much higher. So even if the proportions of diets is small, in which there are salmon, the sheer abundance of fish indicates that more fish would be eaten by those predators.
Go ahead and ask the question. Katie?
Oh, hi. Sorry. I didn't know that's how that worked. Yes, I typed in the chat. I was wondering if you think the catfish might have the highest proportion because they also eat maybe dead or injured ones on the bottom in addition to live ones hunted from the water column.
Yes, that's definitely a possibility. So they definitely do eat a lot of detritus and other things off the bottom. So they could be incorporating dead fish from the bottom. The other thing I noticed from dissections is that the anatomy of the fish is actually a little bit different too. Catfish have much bigger gut content, and so I think there needs to be a little bit more study looking at the potential retention time of genetics in the diet. Because they have a much longer gut, potentially they could be in the system a little bit longer as well.
Hey, I was curious about why some predators would be distributed uniformly while others would be patchy. And I was curious from the predator's perspective, maybe if you're typically if you're distributed uniformly, maybe you minimize competition. But if you're distributed in high densities, is there any chance of like a synergistic effect? Say one predator attacks salmon in a safe place, but then forces that salmon to flee to a more dangerous arena. There's some kind of positive, I guess, synergy of having higher densities. I was curious if you could comment on that.
Yeah. So I'll see if I can address that. There's definitely very different life histories between largemouth bass and straight bass. So straight bass are an anadromous fish, and they can actually move quite freely between salt and fresh water, so that they can move out, to the bay and potentially to the marine waters and come back in. They spawn in April, which is during the smolt emigration. So they're coming in kind of in waves right at the salmon kind of peak time. And they're also kind of a schooling fish, so especially the smaller, they actually will call them schoolies. And so they, will kind of go through like a wave of locusts and kind of annihilate everything in their path, potentially, whereas a largemouth bass is a strictly freshwater fish and basically sets up territories, especially during spawning time, where they have their few hundred kilometers where they like to spend their time. And so the largemouth bass are also more associated with shore habitat, so they like to hang out in the rocky or vegetation habitat on the sides, whereas the largemouth or the striped bass are typically found more in the open water channel habitat as they're kind of cruising throughout the system. So with all of those different factors, you can see how the different species could have dramatically different effects on the salmon smolts as they're going through. But I do see how there could be some synergistic effects between, if you have high populations of striped bass interacting with the populations of largemouth bass, and how they might, be more than the sum of their parts against salmon smolts.
Based on your hydroacoustics work and other studies of the predators, are there perhaps refuges that Chinook might be able to take advantage of during migration?
Yeah, so from the 25-kilometer reach that we looked at, there were very little refugia for the fish. There is some aquatic vegetation throughout the system, but it's pretty thick, and so they probably don't do well hanging out in the really thick, I forget exactly which kind of vegetation it is. I think pretty thick milfoil. And on top of that too, there's also water hyacinths. So these are these floating mats that are going throughout the system. And so those actually provide great habitat for largemouth bass. So you have these floating structures basically for largemouth bass habitat. The system is pretty shallow, between a meter up to about 11 meters deep in the deep scour holes. A lot of it's very channelized and levied, so there's not much refuge for the salmon, at least in this section of the river.
Yeah, I was just curious. So your study was during severe drought years. I'm curious if you could speculate about, the effectiveness of predators when it's, maybe less of a drought situation.
Great. So there have been some studies looking at survival of salmon smolts in high flow years, and they show that they do survive much better in high flow years. What the exact cause of that is, if it's just a dilution of predators, or if they're able to move through the system a little bit faster, that might be. So we have money to continue this work this spring, and we're gonna. So this focused on this one little section of river, and this year we're gonna try and go to multiple locations throughout that whole southern delta and get a lot bigger range. And there's been a lot of rain, at least very recently, in Northern California, so likely have much different conditions this year. So we'll be able to compare between the drought and the flooding conditions.
I had a question. If you have the data about the percentage of salmon that are in the diets of these certain types of fish, and you have estimates about the total numbers of those fish in your study area, do you have any estimates about the total population impact on the salmon?
So we played with that a little bit, so Cyril Michel is the person who's really led the effort for the bioenergetics stuff, and we didn't feel comfortable sort of extrapolating out too much farther than our system, especially for the straight bass, because they're like I said, moving around throughout the whole system. So potentially we could do that with largemouth bass if they're pretty consistent in numbers. So I think we'd need a little bit better estimates of population sizes throughout the whole delta to try and to get that impact. But that would be a great thing to have, for sure.
I've got a question from a user online that we can't unmute. Can you explain how you get from frequency of DNA in the diet to number of smolts eaten? My understanding is that the DNA in the stomachs is more of a presence-absence than a % of diet, so don't see how you get to absolute estimates.
Great. So, again, I did not lead this effort. This is Cyril Michel who's done this, so he can really provide the specifics on exactly how we made those estimates. It's correct that it is just a presence-absence in each diet, but we can get a confidence interval of the proportion of diets for each species. So we can estimate the amount, the proportion of the population that are consuming smolts within a 2-day window. So we are definitely making some assumptions, but yeah, and I'm sorry I don't know the specifics about that. But if you want to email me or Cyril Michel, we can provide you with a few more details in the future.
And there was another question here. Can you discuss some possible correlations among your NVTOL variables and whether there are some associations that might not be mechanistic? For example, the higher predation at lower temps is counterintuitive to the idea that metabolic rate might drive higher predation at higher temps. Is local temp lower in deeper reaches, which might have holes that house a lot of predators, so the low temp relationship is really a proxy for deep predator holes?
Yeah, so just to start off with, we did look at the correlation, the big correlations between all the variables, and we removed anything that was highly correlated. So everything that went into the model was not highly correlated. Temperature, that was interesting that it was lower temperatures. I think one of the things, the temperature, because the system is pretty shallow, there is not a huge temperature gradient even in the deep water. So I don't think it's the deep water that was causing those temperature differences. I think that was, we had a few days where it was kind of stormy weather and we were out sampling. And so I think it had to do probably with that for some reason on that night where it was a little bit cooler, we had higher predation rates. In the system, we're really actually guarding against high temperatures. So in the system, the temperatures get above 23 degrees C in May. So these are basically lethal conditions for the salmon. In 2015, at the end of the season, we had temperatures up to 27 degrees C, which basically would just kill a fish, the salmon, the instant you put it in the water. So I think the temperature range that we see, and the reason we see lower temperatures is because we're dealing with the extreme highs, and so when we have a little bit lower than that, for some reason we're seeing that relationship.
Okay, one more question from online. When were the population abundance estimated for STB?
So the plots that I showed were, I believe they were 2015, right? I think I only showed 1 year, but so 1 year for striped bass, and so I believe in the second year it was similar in the sense that there were higher numbers of striped bass at that removal 1 location. So there were similar trends, but again, there was widely big differences between study reaches throughout the system.
You probably get this question a lot, but I'm wondering if you have a good feeling of how much the tether influences the natural migration behavior of the fish.
Yeah, so these are definitely, you know, influencing the, it's heavily dominated by wherever that float is going, the smolt is going along with it. Although from the camera footage, it's actually pretty surprising how natural the smolt looks. It's not being dragged. It's not just flapping around trying to get off the line. So it actually is a fairly, you know, what you might consider a normal swimming pattern. I do think it would be good in the future to try and maybe compare looking at fish on tethers compared to fish not on tethers and things like that. But there's definitely there's got to be some influence of being on that tether.
We'll give a last thank you for Joe in just a sec. We are going to go to lunch after this, so anyone is welcome to join us. I've got room in the car. And if we can thank Joe one last time.